Welcome to Nucleic Acids Part 2. In this tutorial, we'll look more closely at the structure of DNA. Let's get started. Alrighty, so the nucleic acids are basically polymers of nucleotides. What happens is the nucleotides are joined when the phosphate of one nucleotide joins the 3' alcohol of the other nucleotide to form phosphate ester or phosphoester bonds. All right, so let's, here it's explained to us, but let's connect that to the actual structure. So we're going to look at a trinucleotide, ACG. So the first thing to note is that we read them from the top down. And so this is described as the 5' to the 3'. So we start at the 5', and remember that that was where the phosphate group attached. And then we will end at the 3', and that's where the links occur between one nucleotide and the other, right? And so the other one, and then just since we're talking about the sugar is very busy, right? At 1', that's where the nitrogen base attaches. So out of the 5 carbon atoms on the 5 carbon sugar, 3 of them are very busy. All right, so we look here and we see there's the 5'. So this is the start of the sequence, right? So 5' starts the sequence. Now, every nucleotide has a phosphate and a 5 carbon sugar. So we see that the nucleotides are classified by the nitrogenous base. Okay, so here we would have the first nucleotide, the phosphate group, and the sugar and the nitrogenous base, and then here is the linkage, right? So this would be the phosphoester bond or phosphate ester, two ways to say the same thing, right? So here is our first nucleotide that we classify as adenine. Now this process is going to repeat over and over. So we look at this phosphate group right at the 5', and there's the 1', where the nitrogenous base, all right? So then we have another phosphate ester bond. And we see that that phosphate ester bond at carbon 3 is the link to the next nucleotide. So we can go in here and we separate the nucleotides at those phosphoester bonds. So then this would be cytosine. And then last but not least, we bring in the third nucleotide, and right with the phosphate group linking from carbon 3' of the top nucleotide to carbon 5' of the bottom nucleotide, guanine in this case. And then here we have the 3' ends the sequence. So here is here would be the nucleotide guanine, right? So you want to be able to circle the individual nucleotides, recognizing the role of the phosphate ester linkage to join the two nucleotides together, and recognize that we always start at the top with the 5' of the sequence, and we read down and we end with the 3' that ends the sequence. Now, we only have 4 nucleotides. How can we get so many different genetic variations? Well, the number of nucleic acids that can be formed from 4 different nucleotides is 4 to the n. So just using these 3 nucleotides right here, 4 to the 3rd is 64 different possible nucleic acids. And DNA is composed of millions of nucleotides, so that explains the wonderful diversity that we see among all the different creatures on this planet. Okay, now now that we understand how a strand is hooked together of nucleic acid, we'll go into the actual structure of DNA. So DNA actually has 2 nucleic acid molecules that twist together around each other to form the infamous double helix. It's important to note, right, so it was back in 1953, Watson and Crick were the ones that proposed this structure, but the person that often, she's getting more recognition now, but her name was Rosalind Franklin, and she was the woman who actually collected all the X-ray diffraction data that helped Watson and Crick get to their discovery. And so it's important to acknowledge her contributions as well as theirs. And so now let's think about the nucleic acid structure and how does it create this double helix, right? Well, there's the hydrophilic backbone. So this is where we have the sugar, right, and the phosphate. So what do we know, right? Why would those be hydrophilic? Well, sugars love to H-bond with all those hydroxyl groups, and the phosphate group is ionic. So, right, so this creates the backbone or the ribbon, right? So creates the this water-loving backbone. And so symbolically, you could kind of think of it as like the sides of the ladder, right? So the hydrophilic backbone is like this. But then, of course, we're going to need to twist it, but that'll come soon enough, right? And so, right, because they're water-loving, they want to be on the outside, right, on the outside to interact with our aqueous bodily fluids. Okay, so now we need the hydrophobic part. You could think of this as the rungs on the ladder. So we would put those here. All right, so that would be the hydrophobic nitrogen base pairs, right? And so the beauty is that the nitrogen bases, right? They can H bond with each other, and this is perfect. It adds enough structural integrity to actually form the double helix, but because these are IMFs, not true bonds, it allows for opening and closing. So H bonds are like the Goldilocks, right? There's enough attraction to form it, so there's enough attraction, right, to form the helix, right, but they're able to open, but able to separate. And we'll talk about why that's important very soon. Okay, so we've talked about it in a description. Now let's actually look at the structure, right? So here we see the hydrophilic sugar phosphate, right? That's the backbone there. And then we see the nitrogenous base pairs forming the rungs to the ladder. And then inside here, right, this is representing, right, the dashed lines are the H bonding. So if we see here, let's see, this is an adenine, and this is thymine. And so it's important to note that adenine and thymine they will always form complements to each other, and we can see how structurally they link together with two H bonds. And then if we look at guanine and cytosine, they also always form pairs together. So we'll only see adenine with thymine and guanine with cytosine. We never see them mix the other way. And here we see that there's the ability to form three H bonds. And so if we look here, for example, here's adenine, here's adenine to thymine right there. So here it's shown in the schematic, and this is what it would look like if we got down to the atomic level. And then here, let's see, there's guanine and cytosine. And so this is what it looks like symbolically, and there we see it at the molecular level. Alrighty. Okay, so we've looked to see how the nucleotides bond together to create the nucleic acids. And now we've seen how the nucleic acids come together to form the double helix that we recognize as DNA. And remembering that the nitrogenous bases are restrictive, that it's adenine with thymine and guanine with cytosine only. And now we'll look at the higher order of DNA. Right? So DNA is packaged into chromosomes that are found in the nucleus of the cell. Right? So the chromosomes are highly compact structures. They include both the DNA and proteins. Right? So what are these proteins? These proteins are called histones. And so nucleosomes is a lower level of structure that creates the chromosomes. So basically what happens is the DNA gets wrapped around these proteins, the histones, to create the nucleosomes. And then the nucleosomes coil upon themselves to make the chromatin fibers. And then the chromatin fibers wrap upon themselves to create the chromosomes. So we have it in words, and now let's look at a diagram of the higher order. All right, so this is where we started, right? Here is the double helix of the DNA, two strands of nucleic acids, right, that are polymers of nucleotides. And then here, these small circles are representing the histones, which we know are proteins. All right, so we can see that the DNA curls around the histones to create the nucleosomes. And then the nucleosomes curl on themselves to create the chromatin fiber. And then the chromatin fiber curls up on itself to make the chromosome. All right, so you want to understand, make sure you understand the different components that go into creating the higher order structure of a DNA molecule. And now let's look a little more closely at, we know that DNA holds the genes or the instructions for synthesizing proteins and enzymes. So let's look a little more closely at that for a minute. All right, so there's been a lot of talk, right? The genome is the complete sequence of bases of DNA distributed over the 46 chromosomes. And a gene is a segment of that DNA, right? So the gene is where we have the actual instructions for making the protein. If we look back to our introduction, we talked about DNA as having the instructions, and those instructions are called a gene. So when a gene is expressed, right, so gene expression, right, that's what prompts, right, prompts the synthesis of proteins. And in many cases, these proteins are enzymes. Right? So something happens in our body and we need reactions to occur, then we'll have gene expression to synthesize the proteins, which can then catalyze the reaction as enzymes. So just to give you a sense of how complicated this all is, here is an example of the information we've learned about chromosome 6, right? And there's just, it's overwhelming, so if this is curious to you, you can read some more information. But now let's link this to what we've learned about DNA and chromosome structure, right? So here's a representative of a chromosome. And then when we look inside, we recognize that we see, right, we unravel the chromatin fiber and unravel the nucleosomes. We get down to the double helix of the DNA. And then within that, there is a gene, right? There is a sequence. Here it is, right? There's the nucleotide sequence, which creates a gene on a single strand of the DNA. And remember that that nucleotide sequence is then the recipe for how to make a protein. And remembering that, and so then when we see a G or an A or a T, remember that that represents one nucleotide and that they're characterized by their base. Okay. So let's go into a little bit about how gene expression works, or, well, right here we'll just look at DNA replication. We'll look at gene expression in a future video tutorial. Alrighty, so if we look, for now let's look at DNA replication. Okay, so every person starts life as a fertilized egg, a zygote, which contains the entire human genome. This single cell grows and divides to become a human being with about 1 trillion cells, right? So how does that happen? Well, the DNA is going to need to replicate. Now this is definitely, right, an anabolic process. So we can think of it as endergonic, right? And that it needs, right, it's going to need ATP. Needs energy. Alrighty. So the first thing that needs to happen is we have to unravel this supercoiled DNA to expose its double helix, right? Think about that, right? It coils up on the histones and then it lots of coiling happening. Alright. So the enzyme here is helicase, and then of course we need the ATP. Oops. There we go. All right. And we recognize, right, ACE, right, this is our enzyme, and it has a very logical name, right? We're opening up the double helix, so helicase, and we need energy. All right. So interestingly, each of the 2 strands gets copied, okay? So if we look up here, we would see this as our original strand. Our original DNA. All right. Now, down here, this we call the replication fork. So we have to split it apart. So the helicase is coming in and splitting apart the 2 strands. And so there's the fork where the reaction still needs to occur. Now, if we follow these strands, if we look here, we see there's the 3'. If we follow it, we recognize that this is the parent strand. And so then what happens is there is a second enzyme. And so if we look here, we see this is the daughter strand. Okay, so here, creating the daughter is DNA polymerase. Oops. I messed up my spelling. Here we go. Polymerase, right? So once again, we recognize that this is an enzyme, and it's creating a polymer of DNA, right? It's forming the daughter strand, right? So, so DNA polymerase, right, forms the daughter from the parent. And this is amazing. This happens at a rate of 100 nucleotides, what do you think, per hour, per minute, per second, right? Amazingly fast. Okay, and it's happening simultaneously. So if we look over here, you should be able to identify the parent and the daughter strands, right? So we can see here, this will be the parent, and this is our daughter. Okay, so the main things we want to know about here are that the helicase is what is separating the two forks. DNA polymerase is forming the daughter from the parent. Don't worry about the numbers, I just think that they're really amazing. And then you might be thinking, 100 nucleotides per second? Oh my God, how many mistakes are made? Well, the mistakes, right? Mistakes basically lead to mutations, which we'll talk more about very soon, right? Even at this amazingly fast rate of 100 nucleotides per second, there's only one mistake in 10,000. And then the system does have proofreading. So after proofreading, the overall process is only 1 in a billion mistakes. So thank goodness for that. If it wasn't such a good system, we probably wouldn't be here to talk about it. Alrighty, so that concludes our tutorial on the basic structure of DNA and how it reproduces to make the daughter strands and to form the new molecules. So please take some time now to do some homework to reinforce your understanding.